Thank you so much. I have power and you can see my screen. We're making progress. Okay, let's have the chat open over here. I'm just gonna have to move that so I can see what you guys are saying. Are you ready? I've got more viewers doing fussing about trying to get my computers to work than than teaching biology. Now, I'd like to connect my mouse because otherwise I'm going to keep grabbing my mouse and it's going to keep operating the other computer that's right next to it. Okay, let's go. DNA, genes, and chromosomes. So if you are one of our subscribers, this is our content guide overview page. So you'll see, you know, if you want to go change your email address or your account settings, you want to check your payment date, that's where that is. Little My Courses button will show you all the courses you're enrolled in. This is where our live classes take place. If you are one of our subscribers, I'll send you an email to update you on what's going on with our live classes this week. And then a button to the exam guide, how far through the year we are, although that's kind of gone to pot a little bit right now. And then this is your traffic light progress bar, which tells you how many are sort of red, amber, or green over here. So you can also do a searching page on this page over here. And you can do DNA, if I can type genes and chromosomes. This is the lesson that I wanted to go into. So I'm going to sort of go through the notes with you on screen because neither of my computers want to share my iPad today, which is, so you can see how I've used the traffic light system there, those red and green dots. So let's go through this. I should be able to make this full screen, should work. Are you gonna see that? That's kind of weird. I've got a second, kind of like a second screen. Okay, so DNA, genes, chromosomes. As I'm just gonna check. Okay, 720 should be fine for streaming. I'm just gonna do this in sort of sections. What can you tell me? In fact, probably the best thing to do. Oh, no mouse. Okay, what can you tell me about eukaryotic DNA? How is it different to prokaryotic DNA? It's quite common, this question. Now that we're ready to go. Sorry about the wait, everyone. There's only wasted 20 minutes of your life, but it's not like we're all that hectic busy right now. There's lots of time to kill, so I do apologize. So what can you tell me about eukaryotic DNA? In nucleus. My chat is way over here now, so there's not a lot I can do about that. Maybe if I shut these things on that chair. Disconnect my fancy webcam. Okay, and it's a bit closer to the camera. Okay. Linear associated with histones found in a nucleus. Yeah, very nice. Longer. Yeah, there's more DNA in a eukaryotic cell than there is in a prokaryotic cell. So I'm not looking for differences between the cells. I'm looking for the differences in the DNA between them. Prokaryotic, so we can then flip reverse that. So a lot of these are like the attribute of one is kind of like the opposite of the other attribute. So if DNA in eukaryotes is linear, then DNA in prokaryotes is circular. It's a loop. It's a single, sometimes called a single chromosome. What else can we say about prokaryotic DNA then? There's no histones, not associated with proteins. Histones are the proteins around which the DNA supercoils. If you're doing some A-level sort of ties into that, which it's not all specifications, but you can get some epigenetic changes by affecting the acetylation of those histones will effectively, it can affect the way that genes are expressed, which genes are turned on, which genes are turned off, and they are heritable changes. But that's epigenetics, so that is not for everybody. I'm not going to delve into that right now. Okay, so let's make some notes on this. Oh, mouse. Maybe if I turn it off and turn it on again. I might have to just disconnect from it over here. You know what? You know what's happened to my mouse? The battery's died. Probably charge it once every 6 months, and just today's the day. You know, I think it's going to be that way. Okay, back to the old trackpad. So if we put that in note form, we can say, yeah, it's long linear. It's associated into a double helix. So this is our double helix here. And if we were to then represent that double helix as just a line, the red dots being the histones, the proteins, and that's where you get this just coiling. The helix are just coiled around these histones to form super tight coils. It's found in the nucleus. It's folded around the proteins called histones, and then it's folded into chromosomes. Yeah, okay, you could say a nice one about having introns. Yeah, you can say that for sure. You could say that prokaryotes do not have introns, or the eukaryotes do have introns. There's some interesting ways. In fact, I was asked a question on my course about that just the other day. The question was about a bacterium, and it was about bases being removed from the DNA, and it said do not include references to splicing. And obviously splicing is the process during transcription where the introns are removed, but because it was happening in a bacterium, it was just assumed knowledge that that would not contain introns. This student was asking me, why can't I say introns? It's totally a valid answer to this question, and it would have been if it had been a eukaryote, but because the context of the question was a bacterium, it was assumes you know that it's therefore not got introns, and therefore there is no splicing that takes place. So you've got to be really on it with these every time you're analyzing that question. Oh, this is a prokaryote, therefore this is what's going on in terms of. Is that super fuzzy for you guys as well? I'm just going to refresh. It just seems to be that it's going to. No, it's visible. I think it was just a bit of lag on mine over there. The definition of an intron is the non-coding sections within a gene. It's inside. Obviously, there's non-coding regions between the genes, but within a gene, it's the non-coding sections within a gene. They're not coding for amino acids. Okay. Prokaryotic DNA is pretty much, on the other hand, DNA shorter, less of it in general. There's no proteins, no histones, no nucleus, and there are plasmids. So plasmids, again, double-stranded loops of DNA that are separate from that central circular piece, the master piece of DNA in the cytoplasm, and they are free floating in the cytoplasm. They can be passed between different bacteria, that's how they can pass, like antibiotic resistance. Because what is a species? I'm just going to ask you, what is a species? We looked at this in our sessions a little while ago. What is the definition of overlapping? Maybe non-overlapping. There's 3 ways that we can describe DNA. It's actually coming up on my next piece, is that we've got DNA as universal. It's common to all organisms that we know about on Earth. Universal. The code for, CCG, if that codes for glycine, then it codes for glycine in humans, it codes for glycine in fruit flies, it codes for glycine in prokaryotes. It's also non-overlapping, which means those bases CCG, the next base could be AAT. You can't. So if this is one codon, if I do like this, and this is another codon like this, when one stops, the next one starts. There's not any backtracking over each other. It's non-overlapping. It doesn't do this. It's like one codon, then another codon, then another codon, and they don't overlap. Yeah. So definition of a species: organisms that can produce fertile offspring. What's the problem with that in bacteria and prokaryotes? How do they reproduce? They're not reproducing with each other. They're reproducing asexually by binary fission. Binary fission is only a key term that AQA students need to know. And therefore, what constitutes a species of bacteria, because they're not interbreeding to produce fertile offspring. So there's a bit of the biological species concept, which is what that is a bit flawed when it comes to asexual organisms that reproduce asexually. So, but bacteria can get over this problem of basically everything is being genetically identical by passing plasmids between themselves. And these plasmids sort of encourage a bit of genetic variation, and those useful genes could be antibiotic resistance would be a classic one, but it could be genes for more or less anything. It's a gene. It makes codes for protein, and you can pass them between each other. They're generally a little bit reluctant to take genes from somebody else. It could be dangerous, right? You're taking on a gene, you don't necessarily know the impact of that gene might be, which is why when we come on to the inserting genes, which we're going to be doing in the session after this, encouraging a microorganism to take in a foreign plasmid isn't that the take-up rate is quite low, which is why we need to use marker genes. But we'll come to that later on. Okay. Excellent. What else maybe we'll go through it a bit more verbally, and I'll just share the note at the end. The mouse is empty of battery. Maybe that's a nice idea, and then you can just pause the video during the recording. We can do more of a bit of sort of discussion during the session. So what can you tell me about the DNA? I've seen this on a couple of mark schemes actually in the new spec. Which organelles in eukaryotes also contain some of their own DNA? Mitochondria and chloroplasts. Exactly. So these previously were free-living prokaryotes of some kind that were engulfed in a process called endosymbiosis. They just enveloped them, and instead of eating them and hydrolyzing them, a bit like phagocytosis, they was like, we can do a bit of an exchange here. So chloroplasts obviously are photosynthetic organelles, and mitochondria are basically the site of aerobic respiration, and they're making ATP. So they have, what would the genes within a mitochondrion, what are those genes likely to be associated with? What might be some examples of the proteins that the genes in mitochondria would make? And same for chloroplasts. So those genes in mitochondria exactly are going to be the ones that are maybe going to be making some enzymes in those biochemistry metabolic pathways that do aerobic respiration. So there's every reaction involved in those aerobic respiration reactions are controlled by enzymes. We don't have to learn any of the specific ones. There's a whole bunch: phosphofructokinase, succinate dehydrogenase. Like, it's basically a whole bunch of enzymes. And so the genes in the chromosome, in the DNA, which is basically exactly the same as far as we're concerned, it's exactly the same as prokaryotic DNA. Single loop. You just apply your knowledge of prokaryotic DNA to the DNA that exists inside of the mitochondrion or the chloroplast. Same for photosynthesis, or the genes inside the chloroplast. They're going to be enzymes involved in maybe light-dependent reaction. It could be maybe even ATP synthase. I don't really know what they are. You're not expected to know what they are, but you would be expected to give some suggestions. So I think any sensible suggestion of the proteins which are directly associated to the reactions that are taking place inside of a mitochondrion or inside of a chloroplast is totally fine. Do any of those proteins enter and leave? Possibly. I don't really know. And you don't really need to know either. Okay, where do we inherit from whom do we inherit our mitochondrion from? Yeah, well, I mean, because they're containing DNA, there will be those proteins are manufactured. There is transcription and translation of the genes inside of the mitochondrion and the chloroplast, which tells us there must be some transcription of those genes. You would also assume that there's no introns in there, and that therefore they make messenger RNA, and that they do synthesize those proteins. Quite the mechanism for that, I don't know. Whether the RNA leaves the mitochondrion, goes into the cytoplasm, uses the ribosomes from the eukaryote, and then comes back in, again, you're not expected to know. You would just assume it happens the same way as it would do in a prokaryote. There's from our mother. Yeah, why only from our mother? Because the egg contains the mitochondria. The sperm does contain mitochondria. EdXL students need to know the structure of the egg and sperm in a lot more detail than anybody else. You have the head of the sperm, and the midsection of the sperm is where all the mitochondria are, but there's no mitochondria. Basically, in the head portion of the sperm, you have the acrosome, which is a little bag of hydrolytic enzymes, which it is going to use to digest its way through the zona pellucida on the egg, basically, the outer layers of the egg. But once it's only basically going to insert the DNA from the sperm into the egg cell. It's the only thing that it contributes. And so all the other cell organelles, mitochondria, ribosomes, all the other organelles come from the egg, or therefore from your mother. So that's sneaking in has been an AQA question that doesn't require you to know that, but basically requires you to be able to figure that one out based upon the information in the question stem. Okay. Yeah, I'd say that most other people can hear me. So if anyone else is getting systematic auditory problems, then let me know, and I will. I mean, I think my microphone is working pretty well. Okay, we just recap what is the gene then. So a gene is a sequence of DNA that codes for a protein or a polypeptide. Gene locus is the location or the position of a gene on a chromosome. Okay, let's go over what is the difference between what would I call a sequence of 3 bases in DNA, messenger RNA, and transfer RNA. What would be the terms I'd use to describe the sequence of 3 bases that code for a amino acid? Okay, so on DNA, what have we got? On DNA, I mean, you are allowed to say codon on some mark schemes. I steer away from the use of the word codon with DNA. DNA, I would say triplet. Messenger RNA, I would say codon. And transfer RNA, I would say anticodon. The reason I don't use codon for DNA is because it's all too easily confused with messenger RNA. For me, the codon is on messenger RNA, and then the triplet on the DNA that codes for that codon, I call the triplet. DNA triplet, messenger RNA codon, transfer RNA anticodon. So if you stick with that language, it's easier to make fewer mistakes. You'll make fewer mistakes, basically, if you use that language. Okay, so we've talked about a little bit about non-coding DNA. Non-coding DNA is DNA that does not code for a protein or a polypeptide or for functional RNA. Sometimes you make RNA that interferes with things elsewhere, and only AQA students need to know about siRNA and stuff like that. You would consider the DNA that codes for siRNA to be functional because it is doing something. There's plenty of DNA between the genes. So you've got gene and then DNA, and then gene and then DNA. So I mean, like, let's say on my Apple Pencil here that this orange Mankey finger grip is a gene, and that the silver ring on the end here is a gene. And then between them, we've got all this white stuff, which is DNA, but it's not coding for a gene. So this could be our non-coding bit between the genes. Okay. Within this, you have a whole bunch. It's very common. So you're going to have various, what we looked at in our live class yesterday, year 13, and have various palindromic recognition sites in here, just dotted around. They're just going to be everywhere. There's going to be little palindromic sequences here, which would be active. The enzymes, restriction enzymes active site will be complementary to those, so that it could then, if you've got one here and you've got one here, you could chop and you could chop, and then you could isolate this gene in the middle here. You've also got, what else do we have between the genes? We've got lots of non-coding stuff there. What have we got? Non-coding stuff between the genes. And you're all going to call these slightly different things. AQA have a specific term for it. I can't read that word. If DNA is, it's too small for me to read. Okay, exons, we used to call this junk DNA. That was a term for it. But if we take it away, organisms don't work properly. So I don't think it's useless. So I think the term junk DNA is a little bit out of fashion these days. Okay, I'm talking, but this is the white bit here is between the genes. The orange bit is a gene, the silver bit is a gene. So this is between the genes. So it's not an intron, because an intron would be inside the gene. You've got packing DNA. This is more to do with DNA profiling or genetic fingerprinting. So this is a year 13 topic that I'm angling at here, but they would like you to know that these things exist in the AS level, but you don't need to know. I'd suggest that I'd say it's probably your volume rather than me. I think there's most people are able to hear me. Possibly you just muted it. Any year 13 students here? RNA is the polynucleotide, ribonucleic acid, but there's different types of it. It's folded in different ways. So transfer RNA, you have this sort of clover-shaped, single-stranded, but folded, containing hydrogen bonds. Messenger RNA is single-stranded, but linear, no hydrogen bonds holding it into a certain shape. And there's other types. There's, depending on which example you do, ribosomal RNA. So a ribosome is composed of RNA and proteins folded together. So what do we got? No one's given it to me. So it's basically repeated. There's lots of sections of what you could call junk DNA, but they're repeating sections. You might get AATTTG, 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 and you'll just get 50 of them, the same pattern, and you'll just get 50 versions of it. And AQA students will call this variable number. In fact, the AQA spec will call it variable number tandem repeats. They're not all just tandems, but that's what the AQA spec calls it. Variable number repeats, I think, is what the OCR spec calls it. So you've got this just like random repeats of these sequences that are between the genes. Then we've got the non-coding DNA within the gene. And what are they? You've given me this already. Yeah, okay, exactly, introns. Okay, so if it's within a gene, it's an intron. If it's between a gene, it's non-coding for some other reason. It's a repeating sequence, or it's a palindromic sequence, and there's all sorts of stuff going on between. The majority of the DNA in terms of bases is the stuff in the middle. It's not within the genes. There's only, I don't know the proportion. We've got 20-ish thousand genes in the human genome. And yeah, it's most of the DNA by bases is between the genes. Okay, I'll share my screen and then give you a chance to probably just take a screenshot, and then you can come back. Obviously, if you've got the course, the lesson is DNA, genes, and chromosomes. Let me find it. So if you don't have the course, you can just come back to it. Any questions on that before I skip on through? I'll carry on all the way up to, well, I'll need just a couple of minutes to switch over, change Zoom rooms, and rebroadcast for the year 13 class. But because I was delayed by 18 or so minutes because I don't know what's going on with my iPad. And I don't know why I can't share it off I've been using this laptop that I'm speaking to you on now for the last 5 years. Okay, so what we've got going on over here, we've got our DNA double helix. If we would represent that as a single strand, it folds around these chromosomes, and then these histones, and then these histones basically fold up. And if we were to see it condensed, we'd end up with a chromosome looking a bit like this. Okay, I'm not expecting you to take a copy of all of that down right now. I'll give you a couple of extra seconds if you need to take that shot, and I'm just then gonna 5, 4, 3, 2, 1. Okay. We touched upon a little, we've got, if I can't share my screen, it's going to be difficult. Oh no, I can share questions, but I can't write on it. Let me just try sharing my iPad again. I see no reason why it should not want me to. It must be like a trust permissions thing that it's not enjoying. Let's see what happens here. Just getting black screen from, I've got the live stream open over here. So weird. But I've been using this exact setup for years and years, and why there's obviously a settings change somewhere. Okay, so let's try that another way. We can talk about DNA as being universal to all living things. You can talk about being non-overlapping, that each individual triplet is its own triplet. You can't share a base between two different triplets. And then what's that third word that describes DNA quite commonly? AQA love this the most, although you are expected to know this for all exam boards. Degenerate. And what does degenerate mean? It means there's more possible triplet combinations than there are amino acids. So in other words, some amino acids are coded for by more than one DNA triplet or messenger RNA codon. And this is why it's really useful to be clear with our terminology: triplet for DNA, codon for messenger RNA, anticodon for transfer RNA. Okay, so let's just make sure I think you're pretty, any questions about that is pretty straightforward. But degeneracy, that process by which yeah, you can have multiple, so that classic questions that involve things like that would be: mutation 1 is a substitution mutation, changes a G for an A. It does not affect the tertiary structure, the primary structure, or the tertiary structure of the protein. Explain why. DNA is degenerate. It's a silent mutation. It codes for the same amino acid, does not change the primary structure, does not change the tertiary structure, no change. This is a little bit why, if you're looking to look at evolutionary relationships, if you're looking at, again, with this is a topic we've covered, if you're looking at phylogeny, if you're only looking at the protein, there's various different molecules we can look at to compare how related things are. We can look at the protein, the tertiary structure of the protein, see how well they fit together. So that would be like, does this antibody fit with this antigen? Does that form a complex? And obviously, the more complexes that are formed, the more similar the structure of the tertiary structure of the proteins. Therefore, the more similar the primary structure, therefore, the more similar the DNA. So that by looking at the tertiary structure of the protein, you can infer how similar the DNA is or is not to one another. But you lose resolution if you're looking at the amino acids, because you might get a silent mutation that's taken place in the DNA, which means that there has been a change. So a change has taken place. Those species have diverged a little bit from one another. However, because of due to the degenerate nature of DNA, not every mutation results in a change to the amino acid sequence or the primary structure or the tertiary structure. Therefore, you get most resolution when looking at the DNA. In fact, you get most resolution when you're looking at the non-coding parts of the DNA, because if it makes a change to the DNA in the coding section and it's a negative impact, then it's less likely to survive and reproduce. Therefore, that mutation is more likely to go extinct. What's the proteome? I'm going to go in with a hard one first of all. What's the proteome of an organism? Protein synthesis, not today. There's only, I think protein synthesis probably deserves its own. I will cover it verbally, but I won't sort of delve into the notes. I would say if I asked you then, so pretty close there, yeah, the number of proteins within an organism, all of the proteins that an organism is capable of producing. Yeah, all of the expressed proteins, yeah, but not, I think it's all of the ones that it can make rather than all the ones that it does make, because at any given moment in time, there might be proteins that you are capable of making, but those genes are just switched off right now. So if I said the genome, we would say that is all the genes possessed by that organism. So the proteome is just obviously genes code for proteins. So it's the library of genes that you have is the genome, and it's the library of proteins that you can access is the proteome. Okay, just touch upon a bit of protein synthesis. We've got transcription. What takes place in transcription? Describe transcription, 6 marks. What are the key terms you're going to need to know for that answer. It's quite a common answer. And if it doesn't come up as like a tell me everything you know about it, 5-6 marker, it could easily come up as a, like, those bullet points could be a 1 to 2 marker elsewhere. DNA unwinds or unzips. You can. Controversially, a lot of teachers have said that you're not supposed to say this, but it is in one of the mark schemes. What does the unzipping? DNA helicase. It's in an AQA mark scheme. I don't think it's in any OCR or EdXL mark scheme. I've had teachers tell me that that's not the case, and I just show them the mark scheme and says they give a mark for it, therefore I teach it. That's the mark schemes dictate whether I tell it to you or not. What's true in the real world and what's true in the A-level biology, and maybe not always the same, but I always teach you what's true for the exam. And so they will sometimes, but not always, give you credit for saying that DNA helicase unzips the DNA by breaking the hydrogen bonds. I would use that little phrase because there's often 2 marks involved. If you're talking about DNA synthesis, then there's nearly always 2 marks involved for that. Okay, you get exposed bases. It's correct, but there's probably not going to be a mark for that. Okay, again, just Grace, you said codons in the nucleus. I would just say triplets. I would try to train yourself to use triplets for DNA, because technically, in my mind, you can use the word codon. It's probably not going to cost you a mark, but it could. So I would say triplets, DNA triplets are read. In fact, it's not even really read in triplets at this stage. The triplets really come into it in the translational phase. So I would say DNA bases. What have we got? We've got the enzyme. What's our, and transcription is the thing that we're describing now. What's the enzyme? Again, always going to be a mark for the enzyme. You are required to know it. Bases exposed. You've got pair up. Louis said pair up. A complementary base pairing. Okay, so complementary base pairing, and I would generally give an example there: A with T, C with G, because there's always a mark for that as well. So that's one of the mark points. It's RNA polymerase here. Students get confused with these enzymes, DNA polymerase and RNA polymerase all the time. What are we making? We're making RNA. So the name basically tells us if we're making RNA, messenger RNA, then it's RNA polymerase. If we're making new DNA, it's DNA polymerase. It's the polymer making enzyme, and it's making what polymer? Just that's the DNA or the RNA version. What bond is formed? I don't think I've seen anyone tell me what bond is formed. You've said it joins. You've said it joins RNAs, nucleotides together, forming a phosphodiester bond. That's the you've got nucleotide here and a nucleotide here, and we're making the phosphodiester bond. That is the reaction that RNA polymerase is catalyzing. Okay. So transcription in a nutshell, so we can say DNA helicase unzips the DNA by breaking the hydrogen bonds. We again get complementary base pairing, A with U. I suppose if we're talking about RNA, A with U and C with G. We form, so it's the enzyme is RNA polymerase that forms phosphodiester bonds to add the adjacent nucleotides, RNA nucleotides, if you wanted to say that. This forms pre-messenger RNA or primary messenger RNA. Pre-messenger RNA if you're AQA or EdXL, and primary messenger RNA if you're OCR. They should accept all of them. OCR have just changed the name, what they want you to call it this year. And then we can say splicing removes the introns to form mature messenger RNA or messenger RNA if you're AQA. Mature messenger RNA if you're OCR. So OCR is primary messenger RNA, mature mRNA. And if you're AQA or EdXL, it's pre-mRNA and mRNA. And then what happens to we have our messenger RNA post-splicing, when the introns have been removed, only the exons exist, comes out of the nuclear pore, ends up in the cytoplasm. What process takes place in the cytoplasm? Grace, how does DNA helicase break the hydrogen bonds? I don't know. I was trying to think if I could remember it. or if I ever knew, and I don't think it's something I've ever known. I mean, basically, it will probably be a slight mechanical, I would assume it's a bit of a twist, but basically, how many hydrogen bonds form between A and T, or A and U? This is how complementary base pairing happens. There's 2 hydrogen bonds between A and T or A and U. How many hydrogen bonds between C and G? There's 3 between C and G. So that's how C knows to pair with G, because it will form 3 hydrogen bonds with each other. And there's 2 hydrogen bonds between A and T. And that's how A and T or A and U know how to pair together. Not a big thing. I would guess there is some kind of mechanical action that it's doing as it's rolling along the double helix, but I don't know. And you don't need to know, basically. Okay, translation. Describe translation. More commonly, they're asking now the role of the ribosome in translation, but we can include that in this master question as well. So if we're describing translation, where do we want to go? I'm going to let you lead this in the chat. mRNA attaches to the ribosome. Yep, I'd say that's a good starting point. Or the ribosome attaches to the mRNA. I don't think they'd mark you down either way. You could say which either way around. Ribosome travels along the messenger RNA. Yeah, it's generally going to be fairly near the start codon. Or you can maybe even say it attaches at the start codon. I don't think they're going to mark you down for that. How many transfer RNAs fit inside one ribosome? This is more the role of the ribosome in translation rather than describing translation. You fit two transfer RNAs inside of a ribosome at once. There's often a mark for saying that. Why 2? Well, you've got one transfer RNA and another transfer RNA, each of which carrying an amino acid. And what's the whole point? You want to form a peptide bond between the amino acids. So you need to basically hold the amino acids next to each other, which is why you've got the chain, the long polypeptide chain, which is being formed, the primary structure. And then the new one, transfer RNA, comes in, and you want to basically pop that one on, and then it's going to move along one, and then a new amino acid is going to be brought along by a new transfer RNA over there. Okay, so we can say ribosome attaches to the messenger RNA at the start codon. It reads a sequence of 3 bases called the codon. This is matched with an anticodon on a transfer RNA. Each transfer RNA, there's always a mark for saying each transfer RNA carries a specific amino acid. It's not just random which amino acid it carries. Its anticodon determines which amino acid it can pick up and move or transport or transfer. And then there's always a mark for saying that a peptide bond forms between the adjacent amino acids to form the primary structure or to form the polypeptide chain. The role of the ribosome, that aspect of the question would be that it binds to the messenger RNA, that it reads the codons on the messenger RNA, that it holds 2 transfer RNA molecules in place. It allows the peptide bond to be formed between the adjacent amino acids. What would then happen to that polypeptide chain? How would that protein, let's say it's what's something we might want to export? Let's say it's a protein hormone that you want to excrete into the bloodstream so it can travel around the blood and go have its impact somewhere else. What would happen to that polypeptide chain? Now, we've just finished completing our translation. Stop when it reaches the stop codon. Exactly. So we've just reached the stop codon. That's where we can sort of begin this answer. Where does that polypeptide chain go? Golgi apparatus. What does the Golgi apparatus do? One bullet point for that. Modifies, you could say folds and packages, but modifies, basically it's going to add, maybe could add some carbohydrate side chains or some lipid side chains to it. It's going to help fold it up and assume its tertiary structure. Then it's going to bud off the Golgi body into a Golgi vesicle, be transported to the cell surface membrane where the vesicle will fuse with it and then export that protein as via exocytosis. So the organelles involved in protein synthesis, again, very common question linking to the sort of cell biology and the cell and organelles. Ribosome usually attached to the rough endoplasmic reticulum. Golgi apparatus, vesicle, cell surface membrane. It'll be sort of the process by the movement through which the protein goes in order to be synthesized. Okay, that is that. I'm going to quickly run away and see if I can. I might do a quick reboot on this laptop and see whether the iPad wants to work again, because I want to share some questions with you. Okay. Thanks, folks. Sorry for the delay at the beginning. I'll try and get those. I'll try and get all that nonsense trimmed off the beginning of the recording if I've got any time. I'm like fighting time right now, but I'm sure we'll get it done. Thanks for joining. See you again tomorrow, and I'll let you know when I'm going to do that replacement class as well. So see you then.